So, I'm Sang Hwa Lee, and I have been working in LG for 24 years, and I have been working for developing active ingredients, including the skin lightening agent and skin anti-aging agents. My today's talk is about Transkin, a novel skin penetration enhancing peptide for the transdermal delivery of protein growth factors. Here's my contents: introduction, development of Transkin conjugated growth factors, safety, stability, and efficacy, and finally, summary. I don't think all of you are familiar with LG household and healthcare. So, before starting my talk, I would like to introduce my company briefly and some more detail in cosmetics business. LG Household and Healthcare, shortly LG H&H, was founded in 1947 as the first member of LG Group, and we are the leading company in fast-moving consumer goods industry in Korea. LG H&H has an annual sales of $4.3 billion in 2013, and have more than 6,500 employees. We have three major business domains: home and personal care, cosmetics, and beverage and nutri health. In home and personal care division, we are producing oral care products like toothpaste, hair care products like shampoo, hair conditioners, and hair color dyes, skin care products like body washes, laundry products, paper products like baby diapers, and cleaning and hygiene products. About cosmetics business, I'll talk next slide. In beverage and nutri health business domain, we are marketing world-famous beverage brands like Coca-Cola, Fanta, Sprite, and so on. And we have also several local beverage brands like including mineral waters. It's about the cosmetic business. We offer a wide range of skincare and cosmetic products from mass affordable to prestige. We have in premium prestige brands includes Hu, Ohi, Zoom, and Belief. I heard that the Hu is very famous here in China and beloved by Chinese women. Is it right? So we also have mid to high-end masstige brands, several masstige brands like here. And we have acquired mass affordable brands, the Face Shop, in 2010, and it's also famous in China. I focus on the Ohi brand because it is the host of my main topic today, the Transkin technology. The brand philosophy of Ohi is simple, but not to achieve. It's not too easy to achieve. As a cosmetic leader, offering the best skin science and technology. She's a very famous actress in Korea. She is model for Ohi. One of its sub-brands, the Ohi, the first regenerating line, adopts the stem cell technology in skincare products. We have collaborated with Charles Stem Cell Institute, which is one of the top stem cell research centers in the world. As a result, stem cell formula was developed and introduced to Ohi, the first line as one of the active ingredients. Stem cell formula is consisted of selected growth factors and cytokines secreted by stem cells, which are proven to be helpful to skin regeneration. As I mentioned previously, many growth factors are used in cosmetic preparations for their ability to stimulate cell proliferation, ECM production, and wound healing, totally meaning skin regeneration. Among those, epidermal growth factor, human growth factor hormone, transforming growth factor, fibroblast growth factor, and platelet-derived growth factors are commonly used. Using growth factors in cosmetics, however, have several limitations. Because they are high molecular weight proteins, they could not penetrate into the skin easily, especially across the stratum corneum, which are made of dead cells. So we couldn't achieve in vivo efficacy as much as expected by in vitro data. There have been many attempts to enhance the skin penetration of protein growth factors, one of which is use of liposome, most traditional method, but has a drawback of low entrainment efficiency. Later, PTD, protein transduction domain, was introduced. PTD works on cell membrane well, but does not on stratum corneum, because stratum corneum is made of dead cells. So we thought we need an innovative method. Our choice was phase display. We bought bacteriophage M13 library. The molecular weight of bacteriophage M13 is about 16 million Daltons. Here's the here's the picture of bacteriophage M13, and they have random peptide sequences fused sequences of 12 amino acids fused to N-terminal of gene 3 code protein. Here. The selection procedure is to select the bacteriophage which are penetrated the real pig skin, and peptide sequence facilitating, if the specific peptide sequence facilitate the penetration of bacteriophage, it could also enhance the penetration of growth factors, which is relatively small molecular weight or small size. The selection procedure is consisted of 3 rounds of selection using plant cell and porcine skin. This is cross-section of porcine skin, and this picture is a schematic representation of the selection procedures. DNA sequence analysis of selected bacteriophage were performed, and amino acid sequences were reduced from the DNA sequences. We got 10 candidate sequences. For example, NGSLNTHLAPIL is the most potent one, and we named the name TranskinTM. We have performed the characterization of transkin peptides. We compared the amino acid sequences of transkin peptide and PTD sequences. Transkin peptide is composed of only neutral and hydrophobic amino acids. PTD sequence, however, is composed mainly with hydrophilic amino acid. We can realize that the stratum corneum is composed of dead cells, and the intracellular space is filled with lipids. So the neutrality and the hydrophobicity is very essential properties for facilitating the transdermal delivery of protein growth factors. Transkin peptides could enhance the penetration of fluorescein-labeled target peptide in a dose-dependent manner, contrast to a random control peptide. So we have speculated the mechanism of transkin action mechanism. The way to penetrate the stratum corneum is thought in two ways. One is transcellular route and intracellular route. If the transkin enhances the penetration through transcellular route, the integrity of stratum corneum structure, the structure of lipids and proteins will be disrupted. The other way is through intracellular route. And it is the case, if it is the case, the order of stratum corneum structure will be modulated. And the stratum corneum structure, in changes in stratum corneum order, it could be detected by X-ray scattering and FTIR and DSC analysis. In this picture, we cannot see any changes in stratum corneum integrity. And as I said previously, the stratum corneum, the corneal site consisting of the stratum corneum is made of dead cells and have relatively strong, hard structures. So we can exclude the possibility of being penetrated by transcellular route. And we detected FTIR spectrum, differences in FTIR spectrum and the DSC analysis. So we conclude that the transkin peptide facilitated the penetration of peptide growth factors into skin by modulating the stratum corneum, especially increase the fluidity of stratum corneal lipids. Here's the picture of transkin-treated skin permeation of fluorescein-labeled peptide. Without transkin peptide, the fluorescence was detected only in the stratum corneum and the skin surface. With transkin, however, the fluorescence detected in all layers of skin, stratum corneum, epidermis, and dermis. Sorry. So we have produced transkin conjugated growth factors, EGF and HGH. Expression vectors contain transkin peptide fused to N-terminal of EGF and HGH. They were expressed in E. coli and purified. And biological activity and the skin permeation property was validated. This is schematic presentation. The biological equivalence of transkin conjugated growth factors were validated by N-terminal sequencing. We got same biological activity as their mother forms. This is the case of EGF, as a little bit, the molecular weight is a little bit increased, and the same biological activity. The biological equivalence of transkin growth factors were tested in their ability to stimulate expression of extracellular matrix-related genes by our real-time PCR. They have nearly same activity as their natural forms in stimulating hyaluron synthase 3, laminin, and elastin expression. They enhanced skin penetration of growth factors by about 5-fold when we quantitated the growth factors in the lower part of front cell receiver chamber. This is the cross-section immunohistochemical staining of pig skin treated with human growth hormone or transkin human growth hormone. The brown color indicates the presence of growth factors. There is a great increase in the observed growth factors. When growth factors are extracted and quantitated, the transkin HGH is more observed by 100-fold compared with the HGH itself. Because we do not perform animal experiments, the safety of transkin growth factors were evaluated by in vitro alternative methods. So they showed no cytotoxicity, allergenic potential, and irritants in human skin. Transkin growth factors were stabilized by oil in water microemulsion system, which are usually used to enhance the stability and penetration of drugs in medical approaches. Hydrogenated elastin was used as main surfactant in our system. Stable oil in water microemulsion was produced using three-phase diagram, and the resultant microemulsion was transparent and of low viscosity. The mean size of microemulsion drop was approximately about 25 nanometers. Some properties of growth factors incorporated in microemulsion system was tested. Our system maintained the activity of growth factors, moreover increased the stability by 8 times, and permeation slightly increased. The effect of transkin growth factors contained in cosmetic preparation on human skin was tested in a 4-week clinical trial. Application of sample for 4 weeks resulted in improvement of dermal density, which was evaluated by dermscan-C, and enhancement of skin elasticity measured by cutometer, and finally improvement in facial skin, which was evaluated by Primos. Now, I summarize my talk. Peptide sequence NGS LNTH-LAPIL, which are facilitating skin penetration of protein, was developed and named transkin peptide using phase display method. Transkin peptide was conjugated to EGF and HGH, which results in 5-fold increase in skin penetration, 100-fold increase in skin absorption. They maintain the same biological activities. and showed no side toxicity, no allergenic potential, or skin irritation. To enhance the stability of TranskinTM GF, they are incorporated into microemulsion system, TransZone. Cosmetic preparation contain Transkin growth factor, improved dermal density, skin elasticity, and facial wrinkles. And Transkin growth factors were introduced to OE the first as one of the active ingredients. And thanks for your attention.